Hello. My name is Pat, and I am here to tell you about the phospholipid bilayer. The lipid bilayer is a thin membrane made of two layers of lipid molecules. These membranes are flat sheets that form a continuous barrier around cells. The cell membrane of almost all living organisms and many viruses are made of a lipid bilayer, as are the membranes surrounding the cell nucleus and other subcellular structures. The lipid bilayer is the barrier that keeps ions, proteins and other molecules where they are needed and prevents them from diffusing into areas where they should not be. Lipid bilayers are ideally suited to this role because, even though they are only a few nanometers in width, they are impermeable to most water-soluble, hydrophilic, molecules. Bilayers are particularly impermeable to ions, which allows cells to regulate salt concentrations and pH by pumping ions across their membranes using proteins called ion pumps. Natural bilayers are usually made mostly of phospholipids, which have a hydrophilic head and two hydrophobic tails. When phospholipids are exposed to water, they arrange themselves into a two-layered sheet, a bilayer, with all of their tails pointing toward the center of the sheet. The center of this bilayer contains almost no water and excludes molecules like sugars or salts that dissolve in water but not in oil. This assembly process is similar to the coalescing of oil droplets in water and is driven by the same force, called the hydrophobic effect. Because lipid bilayers are quite fragile and are so thin that they are invisible in a traditional microscope, bilayers are very challenging to study. Experiments on bilayers often require advanced techniques like electron microscopy and atomic force microscopy. Lipids and proteins are the staple ingredients of membranes, although carbohydrates are also important. The most abundant lipids in most membranes are phospholipids. The ability of phospholipids to form membranes is inherent in their molecular structure. A phospholipid is an amphipathic molecule, meaning it has both the hydrophilic region and hydrophobic region. The membrane, therefore, is a fluid structure, with a mosaic of various proteins embedded in, or attached to, a double-layer, bilayer, of phospholipids. An example of a cellular membrane is a plasma membrane, composed primarily of lipids and proteins. Membranes are held together by weak hydrophobic interactions, much weaker than covalent bonds. Most of the lipids and some of the proteins can shift laterally. Consequently, membranes can be considered as fluid. At high temperatures, body temperature, cholesterol makes the membrane less fluid by restraining phospholipid movement. At low temperatures, cholesterol hinders solidification by disrupting the regular packing of phospholipids. However, because cholesterol also hinders the close packing of phospholipids, it lowers the temperature required for the membrane to solidify. Thus, cholesterol is a temperature buffer for the membrane, resisting changes in membrane fluidity that can be caused by changes in temperature.